Somebody, ಯಾರಿಗೂ ಗೊತ್ತಿಲ್ಲ? Okay. So I will tell you. So pure culture means, so if you take any habitat ಅಂದ್ರೆ the place where the living organisms or microorganisms are there. If you take any habitat, so like the forest environment or your home or water or soil or the inside portion of our body or mouth or on the hand or our clothes. So wherever the microorganisms are colonized and growing, that is called as a habitat or niche. So if you take whatever the habitat, in each habitat, there will be so many number of thousands of microorganisms which are habitated and growing there, surviving there. And these thousands of microorganisms, maybe bacteria, maybe fungi, maybe actinomycetes, or whatever the type of microorganism. So within type of different groups of microorganisms, so there will be 1,000 different genus. Not at least, not even 1,000, at least minimum tens of different genus of bacteria, different genus of fungi and other microorganisms will be living or will be there in a particular habitat. Okay, so the pure culturing means separating one genus and one species, like Homo sapien or Bacillus subtilis, separating one species of a bacteria or any other organism, either fungi or either actinomycetes, either algae or anything, separating that and growing that separately in an artificial media without growing any others or without getting contaminated with other species of microorganism is called as a pure culture. I will repeat, growing one single species of organisms, so without getting contaminated with other organisms in the laboratory, is called as pure culture. Pure culture. So, and pure culturing is not easy. Pure culturing was not easy enough because so wherever you want to try to separate one organism from other organisms, so there are no places where the microorganisms are not there. So if you prepare a media and if you just open anywhere, whatever the place, the air touches that one. So if air touches, air contains different groups of microorganisms, all that will grow on this. So it will contaminate. So you have to do a pure culturing in a particular sterile environment or aseptic condition, where sepsis or septic means infection, where you should grow or separate this organism from the sample, whatever the sample you have, in an environment, so where there will not be presence of other microorganism or microbial free area. So there only you can able to pure culture. So can somebody, if anybody knows, can you tell me the place so where we can do this or where we can create an aseptic condition or area without microorganisms? ಯಾರಾದರೂ ಗೊತ್ತಿದ್ರೆ ಆನ್ಸರ್ ಹೇಳಿ.
Vacuum, sir.
ವ್ಯಾಕ್ಯೂಮ್. ವ್ಯಾಕ್ಯೂಮ್ ಅಲ್ಲಿ ನೀನು ಕುಳಿತುಕೊಂಡು ಮಾಡ್ತೀಯಮ್ಮ ಕೆಲಸ? Can you able to work with in the vacuum where there is no air? ಆಮೇಲೆ have you heard of laminar airflow chamber? I think whenever I was explaining you about instrument, I have explained you this. So laminar airflow chamber is a chamber, so where we create microbial free condition. So in that we can separate a individual organism up to a species level and we can grow. So that technique we will learn today, at least whatever the extent you can able to understand, please try to understand this. So first, this is the pure culture, growing a single species of organism. So this one is achieved by separating one cell. So by separating one cell from all other and growing only a single cell. So if you can able to separate a single cell and grow only that cell in artificial media, so only single cell will multiply. So that is pure in itself. It is not getting contaminated or mixed up with other culture because you have grown it from single cell. So that is called pure culture. Okay. So what is pure culture? A little bit about definition. So it is a culture that contains a single known species or type of microorganism. I will repeat, pure culture is a culture that contains a single known species or type of microorganisms. Okay. So here, if you whenever this is a mixed culture. Okay. So whenever you take any sample, you take a water sample, you take a air sample, you take a soil sample, you take a juice sample or whatever the sample, and if you try to grow microorganism, you will get like this. You will get a mixed culture. Whatever the microorganisms are there in it, so whichever can grow in the artificial media where you are trying to grow, all those organisms will grow. So you see here, each colony is, there are few colony which are similar, few colony which are different. There are at least 10, minimum 10 types of bacteria are there in this plate. Similarly in fungi, here below plate, so all the colonies are not same. They morphologically they are different. That means to say that there are more than one type of fungi which are growing here. Similarly here in the top plate you can see that. So looking into the morphology of the colonies of bacteria, so there are many types of bacteria which are present in the plates. So this is a mixed culture. Where, so from mixed culture, you have to prepare initially whenever you culture from a natural sample, you will get a mixed culture. From mixed culture, you have to separate one organisms or one single cell, and then you have to make it a pure culture. For example, so this plate where there are 3 fungal colonies, this is all 3 are identical. So this is a pure culture. So this is taken from this one single colony, this colony. So we have separated from all others and we have grown this. This is a pure culture. Because we are growing one type of from one particular cell or one particular species of fungi, we have grown this. This is a pure culture. Okay. But majority of the times, whenever you do, you will get a mixed culture. From mixed culture, we have to separate. So separate means, for example, when you put a soil sample, you will get a colonies like this. So if you want to pure culture any bacteria, so this is, I think you have heard about serial dilution. So whenever we do a serial dilution technique, here, whenever we do a serial dilution technique, this is serial dilution technique. I think I have explained you. If you have any doubt, you can ask me. So the colonies, each colony, whichever is coming here, we say that this colony, each colony comes from single viable cell. In serial dilution plate technique, we say that each colony here, so these are coming from a single viable cell. Single viable cell means, so this single cell has multiplied and formed a colony here. So this colony is pure in itself. The colony which is, this is independent, which is not mixed with any other colony. So this colony is formed by multiplication of a single cell, which has fallen here. So it is pure in itself. So if we can able to separate this bacteria without getting contaminated from all others, so you can pure culture. So today we will learn so how we can able to separate this organism. It's a simple, but you should know how to do that. Okay. So the most commonly used method, laboratory methods for pure culturing are streak plate technique and pore plate technique. Streak plate and pore plate and spread plate. So both the methods, streak plate as well as pore plate, in both the method, they rely on the dilution of bacterial cells in a sample to a point which a single cell can divide, giving rise to a single colony. ಇಲ್ಲಿ ಸ್ಟ್ರೀಕ್ ಮಾಡಬೇಕು ಅಂದ್ರೆ ಯು ವಿಲ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಈ ಸ್ಲೈಡ್ ಅರ್ಥ ಆಗಲಿಲ್ಲ ಅಂದ್ರೆ ಪರವಾಗಿಲ್ಲ. ಸೊ ದಿ ಮೋಸ್ಟ್ ಕಾಮನ್ಲಿ ಯೂಸ್ಡ್ ಆರ್ ಸ್ಟ್ರೀಕ್ ಪ್ಲೇಟ್ ಮೆಥಡ್. ಕಾಮನ್ ಆಗಿ ಪ್ಯೂರ್ ಕಲ್ಚರ್ ಮಾಡೋದಕ್ಕೆ ನಾವು ಉಪಯೋಗಿಸ್ತೀವಿ. ಫಾಲೋಡ್ ಬೈ ಸೀರಿಯಲ್ ಡೈಲ್ಯೂಷನ್. ಸೀರಿಯಲ್ ಡೈಲ್ಯೂಷನ್ ಮಾಡಿ ಇದೇನ್ ಹೇಳುತ್ತೆ ಅಂದ್ರೆ, ಫಸ್ಟ್ ನಾವು ಸೀರಿಯಲ್ ಡೈಲ್ಯೂಷನ್, ಯು ಹ್ಯಾವ್ ಟು ಡೂ ಎ ಸೀರಿಯಲ್ ಡೈಲ್ಯೂಷನ್ ಆಫ್ ಎನಿ ಪರ್ಟಿಕ್ಯುಲರ್ ಸ್ಯಾಂಪಲ್. ಯಾವುದೇ ಒಂದು ಸ್ಯಾಂಪಲ್ ಅಲ್ಲಿ ಯಾವುದೇ ಒಂದು ಆರ್ಗಾನಿಸಮ್ ನಾವು ಪ್ಯೂರ್ ಕಲ್ಚರ್ ಮಾಡಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ, you have to take that sample, you have to serially dilute, you have to plate it on the petri plate, and you should initially get a isolated colonies. ನಿಮ್ಗೆ ಮೊದಲು isolated colonies ಬರಬೇಕು. ಈ isolated colony ಇಂದ ನಾವು streak plate method ಇಂದ by using a different methods of streaking, so we can pure culture this colony. So ಈ ಕಾಲೋನಿ ಅಥವಾ ಈ ಕಾಲೋನಿ ಬೇಕಾ ಯಾವ ಕಾಲೋನಿ ಬೇಕು ಅದನ್ನ by streak plate method we can pure culture. Okay, that's what this slide says. So this so the most commonly used method for isolation of bacteria or for counting of bacteria even is serial dilution technique. Serial dilution technique is the most common method for isolation and enumeration. Enumeration ಅಂದ್ರೆ counting ಎಷ್ಟಿದೆ ಅಂತ. For isolation and counting of different groups of organisms from any sample. So even this serial dilution technique is also useful in pure culturing. So because when you do a pure culturing, when you do a serial dilution, you will have after doing a serial dilution when you plate, so you will get a isolated colonies which are developed from single cells. So if the colonies are developed from single cells, so this can be easily separated onto other plate and it can be pure culture. I don't know whether I have explained you about this or not. After serial dilution, the plating is done in two methods. So after serial dilution, the plating can be done in two methods. One is, so whatever the method I have explained is called as serial dilution pour plate technique. Serial dilution pour plate technique. So after doing the serial dilution, so we will put the sample, this 1 ml of aliquot into the petri plates. So after putting the sample, this microbial containing sample, we will pour the media. We will pour the media above this, we will mix it and incubate, allow it to grow for the colonies. This is called a serial dilution pour plate technique. We will be pouring the media on the, one second. We will be pouring the media on the sample. Here first we will put the sample. So then we will pour the media, mix it, incubate to get the colonies. This is called pour plate technique. There is another method. So here, so instead of empty with sterile petri plate, we can take the plates which are already having a media, already having a solidified media. After above this, you can transfer 0.1 ml or 1 ml of the aliquot, serial diluted aliquot sample. Then using a sterile spreader, we can spread it and incubate it to get the isolated colonies. So here we are putting the sample above the solidified media, then we are spreading and incubating. So this is called as a spread plate technique, serial dilution spread plate technique. This is serial dilution pour plate technique. Okay, both the methods can be used for getting the enumeration or isolating the bacterial or fungal colonies, and even for pure culture. If you have anybody has any doubts in between, you can ask the questions. Okay. Next, techniques to isolate microorganism in pure culture. So once you have done a serial dilution, so you have got the colonies from a particular sample. So how to get, so say for example in the B, picture B, where you can see, so there are different types of colonies. So from this, so you have to pour pure culture one particular bacteria. How to do that? So this is done by streaking. Just you take a inoculation loop, sterilize the inoculation loop. You have to, without touching the other colonies. So whichever the colony you want to pure culture, just with the end of this loop, just touch it. After sterilization with the inoculation loop, just touch this and streak it here. So you have a chance of getting a pure culture. So in streaking, there are different methods. Okay, why that is followed, I will tell you. So here, this colony contains billions of more than 100 crores of cells. So here this colony contains more than 100 crores of cells, because of which you can able to see it as a colony. If there are one cell or 1,000 cell or 10,000 cells, so you cannot see this colony with your naked eye. Since there are crores of cells accumulated by multiplying, so this is a single, you can see it as a single colony. As per the assumption of this serial dilution, we presume that each these colony are pure in itself, because each colony is coming from a single cell, each by multiplication of a single cell. So if you separate these without getting contaminated from other colonies, so you will get a pure culture. Okay. So here, pure culturing, it is done by after serial dilution, each colony we will be taking and streaking. By streaking, we will get a pure culture. So streak plate method. Once you get a colony, so there are different methods of streaking. So this is called as, whatever the method explained here, it is called as a quadrant streaking. Quadrant streaking means we are streaking 4 times. 4 times we are streaking. That's why it is called as a quadrant streaking. So here, so you take a colony, you take a Petri plate which has these colonies after serial dilution, or any bacterial culture. Any bacterial culture, if it is pure. Otherwise, this is difficult. Otherwise, if you have a liquid culture, this culture you have to do a serial dilution, you should get an isolated colony. From this isolated colony, so you take a inoculation loop in a laminar. This needs to be done in a laminar airflow. All the Petri plates, media, whatever you use, they should be sterilized first. Take a inoculation loop. So sterilize by flame sterilization. So then touch this colony. Touch this colony. Afterwards, so you hold the plate in one hand and you do a streaking. So this is a first streaking. So again, you sterilize the inoculation loop. You listen it carefully. Second time, you sterilize. Don't touch the colony. Just touch this last line. Just touch this last line and do a streaking. Again, sterilize your inoculation loop. Touch this is the second one. So sterilize, after sterilization, touch the second line, this line, and again do a continuous streaking. Again, after doing a third streaking, again sterilize. So again, just touch this line and you do a loose streaking. The third one, you can see this is a loose streaking. So first 3 streaking are you have to do vigorously, close streaking. I will repeat once again. So take a inoculation loop. This is called as a quadrant in streak plate method. There are many types of streaking. The method which I am explaining is a quadrant. We are streaking 4 times. Take a inoculation loop, sterilize. So touch the colony, just touch. Don't take anything, just touch, because if they are coming, they will attach to this one. You will get enough number of colonies. So here, streak it. First, take the plate, streak it as much as possible closely. Sterilize again. So again, by touching this line, streak closely. Second time, again sterilize. By touching the second line, streak closely. Again sterilize, and fourth time, do a loose streaking. I will tell you why we do this one. So this is quadrangle streaking. So this is streaking 3 times. This is called as a, 1 second. This instead of quadrangle, you can streak thrice. 1 second. I want to know if there are any name for this. The method of streaking, quadrant streaking. Okay. I'll come back. So instead of 4 times, similar way, you can, this is a triangle streak also, you can say that, or 3 times streaking. So here what we do is, first, take a inoculation loop. So sterilize it, touch the colony. You are touching the colony only once. So then do a first streaking, close streaking, then again sterilize the loop. Second streaking, second also close streaking, and third one again sterilize and do a, after touching this line, do a loose streaking. This is a 3 times streaking. And there is one more method. So here you can see. So this is a simple streaking, just one streaking. This is a two streaking, one and two. Second one, last one will be usually loose streaking. If it is a one, this is also loose streaking. This is the three times streaking, one, two, and three. This is quadrant. One, two, three, and four. This is quadrant. One, two, three, and four. Each time you have to turn the plate while streaking. Okay. This is three times streaking. One, two, and three. So one, I think the video conference will end now. So you please re-log in immediately once it ends. So this is first streaking, second streaking, third streaking. There is four, I think. We have done four here. So here 1, 2, and 3 loose streaking. I will tell you why we need to do like this. There is another method after quadrant streaking. It is called as the T method of streaking. So you divide the plate. First half you will do only one streaking. Second half you will do another two times streaking. So here you will do larger streaking. So smaller one and loose streaking in the third time. I'll show you this. So here like this. First streaking, take a sterilize the inoculation loop, touch the colony, do a very close streaking until it covers the half a plate. Turn the plate. So sterilize the inoculation loop by touching this, just